Good afternoon, everybody. I am an associate professor at the University of Verona in cognitive neuroscience and neuropsychology. I said this because generally people think that I'm a psychometricist, but I'm not. I'm a neuropsychologist. So I want to stress that because sometimes people are thinking that my field is another one. And I'm working in particular in body representations, in particular in spinal cord injuries, but not only, in neuropsychology and also space representation. And probably for this my interest, I've started to be interested in Bayesian statistics since 2011, when the tools to do Bayesian statistics were very rare. I'm also the secretary of the Italian Reproducibility Network, that is an association, scientific association, with the main purpose to spread open science. Okay, and the next national meeting will be here in Verona in the 12th and 13th of February 2026. So consider you booked on these 2 days that you will come here in Verona for this national meeting. I'm also the teaching delegate of the PhD program in human science here at the University of Verona, and director of a summer school of about Bayesian statistics since 2019. These are just to stress again that I'm not a methodologist, but I'm a neuropsychologist. These are some of my representative works, but in effect, I also have some works in psychometrics. Okay. So let's talk about the content of today. Okay. I will talk about a couple of studies where open science has a strong impact. Okay. The first, both of them are registered reports, and I will tell you what a registered report is. Study one published articles in Cortex. First author is Gabriele Fusco, and the second study is a work that received an in-principle acceptance, but is not published yet because we are collecting the data, actually. So the common aspect, there are two common aspects between these two studies. One is that they are both using techniques that are of a non-invasive brain stimulation, and both of them are registered reports. So open science is at the base of both studies. But what is open science? Okay, so open science, of course, is an umbrella term that will consider these 5 aspects. Of course, open data, this is one of the most important things. So when we collect data, our data are not just our data. Our data are not our data. Our data are data for the scientific community. So they should be shared with the scientific community for everybody, from the raw data, not entirely processed data, to the final data. But also all the our scripts that we use to analyze this data, even the scripts that we use to pre-process data and also to remove outliers and so on. So open source software, this is important, okay? Open materials, we should be as transparent as possible with the open materials, and in principle, we might share even our scripts, okay? And even our experiments in open science. And also pre-registration. Pre-registration is extremely important. It means that basically we are declaring before to do an experiment what we want to do, okay? But in detailed terms, even already saying what I expect to do as statistical analysis, okay? But always remember that a pre-registration is not a cage, okay? It's just a plan, okay? So if, for example, something will be slightly different from what you planned, you can just prepare an amendment with the rationale of this difference, okay, and you can go on with your study, okay. In both studies that I present, okay, there have been some differences from what we actually pre-registered to what we actually did, okay? Because we know sometimes things are complicated and sometimes our plan needs some changes. But a pre-registration at least shows that from the beginning we had a specific idea and we never completely changed everything about our hypothesis, for example. The last aspect of open science is open access. Okay, you see here as the first point of open science. Actually, I think that this is the last point of open science because actually open access gave a lot of power to publishers because at the end we are paying publishers to be published. So at the end we are just reinforcing the publishers, the publishing system that actually pushed the replication crisis. About the replication crisis, I think that all of you knows very well what is the reproducibility and replication crisis. Okay, so here we have an estimation about on different scientists in different fields thinking if there is a reproducibility crisis and if they failed to reproduce an experiment in different fields, and you see here the percentages are very high, but there are also studies that actually tried to replicate, reproduce studies. I think that all of you knows the replication crisis of in psychology, published in this article that was published by the Open Science Collaboration in 2015, that showed that actually only the 36% out of 100 studies was able to be replicated as the original paper. But this is not only related to psychology. It is also possible to see that even in studies, in medical studies, like studies connecting with spinal cord injuries, they have a replication crisis. Only the 17% of studies was replicated. Okay, here we have only 12 studies, but a minority. But also in the reproducibility project Cancer Biology showed that in 112 studies, only the 36% of studies was possible to be replicated. So reproducibility crisis is true, and we should be worried about that. But I think that a lot of people, when thinks to reproducibility crisis, thinks also to bad research practices. Okay, so actually, if I am objective in my study, if I am employing state-of-the-art methods, if I am rigorously collecting the data, if I am applying a correct statistical approach, then why should I care about open science? So if I'm honest, okay, if I'm doing good research, if I'm trying to do what is reported as a gold standard, why should I care to actually pre-register my study? At the end, isn't it something only to limit bad research practices? Actually, it's not. So let's see all these 4 points at the end. So if I am objective, can we really be objective? If we ask to philosophy of science, there is a branch that is constructivism that says that objectivity in science is not possible. It's not actually possible because the scientists formulate the hypothesis, conduct and interpret the experiment. Okay, all of this is done by observers. We also decide how to analyze the data, okay? And we also interpret what we observe. So this is difficult to say I am objective, okay? So let's watch this example. For example, let's say that I am a researcher, okay? I want to do an experiment using, I don't know, the Stroop test, okay, on using reaction times, okay? I have a specific hypothesis. I believe in a specific idea, okay? So these are all the possible specifications of this Stroop task. Here we only have the valid, here only the non-redundant, and here there are my decisions. For example, I would use the Stroop task with words and colors, okay, and I will analyze the reaction times. Here we have two researchers with similar views, okay, and here more or less they are very similar, but then there is a difference, okay? Let's say that here I'm using letters and colors, and here different pairs of stimuli, okay? But always reaction times. Here, maybe we have two researchers with very dissimilar views, okay? So, and at the end, let's say that here I'm just using reaction times with a certain type of stimuli, and here a variation of the Stroop test, and I'm actually using the accuracy. So another researcher is using a completely different way to analyze the same hypothesis. So actually all these visions can be true, can be reliable, okay? So it is difficult to say I am objective. There are so many choices that we do during our planning our experiment that is actually difficult to say I am objective. So let's remove that. It's difficult to be objective. There are so many decisions. So I am employing state-of-the-art methods. Okay, I look to the literature and I want to use state-of-the-art methods. Okay, so let's think to this kind of experiment. We want to determine whether in post-stroke patients with neglect, high level of anxiety are associated with more debilitating neglect. Okay, so we are short of time, so I'm not asking you how many groups you want and everything. I just show you how many differences we can have just modifying these three aspects. So let's say that with the first experiment, I will just use one group of post-stroke patients with neglect. I would never use just one group, but let's say that some of us want to do that. Just the stay-y has a test to evaluate anxiety, and we assess the neglect with the dissection test. Here we want two groups, one group with post-stroke patients with neglect and one control group of healthy subjects, okay? But has test to assess the anxiety, the Beck's anxiety inventory, and here another test, instead of dissection test, we use the Bell test, the campanelle, okay? Otherwise, another way to do always the same thing. We are using three groups, okay? One group of post-stroke patients, one group of healthy subjects, and one group of post-stroke patients, but without neglect, okay? With the lesion more or less in the same area, okay? But without neglect. And here, to assess anxiety, I'm using the hospital anxiety depression scale, okay? Instead of the other two. And here, to evaluate the neglect, I'm using the behavioral inattention test, okay? So, how many different studies could I have with all these differences? Okay, let's watch it. Okay, I can have 27 different methodologies to study the same hypothesis. So it's different, and I mean, all these decisions might be good, okay? Might be decisions that that can be publishable, okay? Because all these tools are able to assess neglect, to evaluate anxiety, and so on. But they are different, okay? So, am I sure that I'm actually using the state-of-the-art method? So let's also remove this part. If I am rigorously collecting data. Okay, perfect. Let's say, let's simulate, we actually are working always with samples, not with the statistical population. So a sample has a random variation. So let's simulate 50 observations with effect size, a strong effect size of 0.5, a coherence D of 0.5, okay? I'm simulating 25 studies. It's just a t-test, okay? Nothing complex, okay? So let's generate the data, okay? The blue points are statistically significant results. I have a strong effect size. The red ones, the only red one is a non-significant result. But let's generate the data again, okay? Here we have two samples that were not statistically significant. Here another one, here another one, here another one, hey, and here three. Okay, so even if I am rigorously collecting the data, my sample is a random sample, so there is variation. So let's remove also this part. Even if I am rigorously collecting the data, I'm not sure that my data will actually represent the statistical population. If I am applying a correct statistical approach, okay? So actually, this is okay. This is another aspect that is extremely sensitive because actually I don't think that there is a really correct statistical approach. Never. There is, of course, there might be wrong statistical approaches, but a real correct statistical approach, as Box said, there are statistical models are always wrong, but some of them are useful. So let's see how we can choose a statistical approach. Well, there is an ancient way to do statistical analysis. We decide the statistical test before to do the experiment. We collect the data and we apply that. Okay, just like that, and we never change anything. Otherwise, there is this most modern way where we actually pre-register our statistical test, pre-specifying everything, and then we apply that. And this is something that everybody can check if we actually have done that. Otherwise, there is this other way to do statistical test. It is also called the researcher's degrees of freedom, okay? Every time I'm collecting a different data set, I'm applying a different statistical test, okay? Sometimes it's not just because I want to push my study, okay, towards the statistical significance. Sometimes it's just that I learn more things, okay? Let's think that at the beginning we can study reaction times with, I don't know, an ANOVA. After a while, during my PhD, in the first study, I'm just applying an ANOVA on raw reaction times. After a while, during my PhD, I learned that the reaction times are not normally distributed, so I put them after a logarithmic transformation, and I'm applying again an ANOVA, and this is another way to do a statistical analysis. Then, at the third year of my PhD, I learned that I can use generalized linear mixed models, and I will use raw reaction times but with an X-normal family distribution. So, three different tests with on different data sets, just because I've changed my vision, okay? Otherwise, there is the fourth way, that is just trying all the statistical tests until I find a statistically significant result. This is, of course, a bad research practice that hinders replicability and reproducibility of research, and this is the so-called phishing, and should be avoided as much as possible. It should be avoided. You should never do that, okay? So we know that data are not passively collected. We choose how to remove outliers, transforming variables, choosing independent variables and confounders. These are all routine decisions for researchers. So from a single data set, at the end, according to our decisions, we can have multiple different data sets, a multiverse of data sets. Then a researcher selects a single data set and we analyze that. So it's difficult to say I am using a correct statistical approach. There is something subjective in this part of our research that is not avoidable, okay? So to summarize, because we fail to be objective, because my methodological choices have an impact on results, because of data sampling variability, and because there are many accepted statistical approaches, we need open science. We need to pre-register, to be transparent on what we are doing, okay? Sharing our data actually increase our trustworthiness, and if another person can analyze our data, can actually say, oh yes, he analyzed or she analyzed data in this way. I've analyzed the data in another way and I found the same results. So actually it shows that your result it's robust, okay? It happened to me, okay? I've shared my data on the Open Science Framework. Someone else downloaded the data set, analyzed that in another way, in a different way, and published an article, a meta-analysis at the end, but also reanalyzed completely my data in a different methodology, with several criticisms about what I've decided to do. But the important thing is that even if he was even with this criticism, he reached my same conclusions. So actually, I showed that my results are robust. He showed that my results are robust. Okay. So now let's talk about non-invasive brain research. Okay, let's start with this first study published in 2024, modulating preferences during intertemporal choices through exogenous midfrontal transcranial alternating current stimulation. Okay, we studied the conflict monitoring. What is conflict monitoring? Each day we need to we face several decisions, and some of them are difficult to do, and this activates our conflict monitoring. Theoretical and computational models show that conflict and error monitoring are based on the anterior cingulate cortex and midfrontal cortexes, okay? And that conflict and error processing are connected with enhanced midfrontal theta oscillations, okay? And we actually found also in 2018 that applying a theta tACS, transcranial alternate current stimulation, to midfrontal areas fasten the decisional process during conflict monitoring. So we apply the transcranial alternate current stimulation. So we wanted to use again this transcranial alternate current stimulation. That is a methodology where we are applying just alternating current stimulation to the scalp, okay? And it should reach the brain. Okay, in this way, it actually synchronizes during the online use of this tool. So basically, we are applying the transcranial alternating current stimulation while people are doing a conflict monitoring task. In this way, we are modulating, synchronizing the activity of the brain with our frequency. Okay. So we had a couple of different questions. So investigate again whether the midfrontal theta stimulation can impact conflict processing, fastening reaction times, and explore behavioral carryover effects of this aspect. Okay, because we actually assessed only during online tests. Okay, and we wanted to be as robust and transparent as possible, so we tried a registered report. Okay, what is a registered report? Is a type of scientific article that is something more than a pre-registration. Okay, we actually pre-register everything of the study, but this pre-registration is peer-reviewed before data collection begins, and there are two stages. A stage 1 peer review, so the peer review is actually only on the study rationale and the methods, okay? And then stage 2 peer review, where that is done after the data collection and the writing of the report, okay? So it reduces publication bias, promotes transparency and reproducibility, and there are key aspects, okay, encourages stronger study design through early feedback. So if you're doing something wrong, okay, a reviewer is saying you that there is something wrong in your study before finishing everything with your study. Okay, so in stage 1 of the registered report, before data collection, we submit introduction, methods, and proposed analysis, and we are peer-reviewed for theoretical importance, rigor, and feasibility. And if everything is accepted, we will receive an in-principle acceptance. Okay. In stage 2, after data collection, we submit only results and discussion. It is impossible to modify introduction, methods, and proposed analysis, but only results and discussion. And reviewers will check if the protocol was followed and the conclusions are supported by data. So if we follow the protocol, we will be published anyway, okay? Even if our results are completely new results, even if we don't find anything, okay? So in this way, we are free from the worry of not getting any result, okay? I've pre-registered my study. It is already accepted, okay? So even if my experiment don't work at all, I will be published. And in even good journals, I mean, this article was published on Cortex, okay? So, and this is extremely important, okay? So our procedure was this one, basically, but I will go back also later, but before to do the stage 1, we started with a pilot study because we didn't know what to expect, actually, okay? So from our pilot study, we collected data from 14 participants, actually, and what they've done during this experiment. Basically, they were stimulated with the transcranial alternating current stimulation during a temporal discounting task. What is a temporal discounting task? A temporal discounting task is a task where they ask you to choose between two decisions that are, for example, having a lower amount of money now or a larger amount of money in 3 months, okay? Each time you can decide between having the money now, so an impulsive, let's say, decision, but it depends, or a future greater gain, okay? That means that you have more cognitive control, okay? So this is a decision that involves conflict monitoring, okay? Each decision will modify the amount of money that you will receive now. So if you choose the future, the now quantity of money will increase. If you choose now, the now quantity of money will decrease, okay? And during this task, people would receive the transcranial alternate current stimulation, okay, for 8-9 minutes. So they've done this task online. Okay. Okay. Okay, they've done this task online. Which kind of stimulation we applied? We applied a stimulation that could that was on the a mid-frontal theta stimulation, theta 1 or theta 2, or a control frequency that is the gamma stimulation. Okay, another frequency basically. Okay. Okay, people came in 2 sessions. So we had a first session that always started with a sham stimulation. So they only had the perception of the transcranial alternating current stimulation starting, but it is not actually working. And then other 2 blocks with, for example, the theta stimulation. And then after at least 5 days, another session that would start with 2 gamma or theta block stimulations, and then a further sham stimulation. Okay, the order of the 2 sessions was balanced, but also the order of the gamma and theta stimulation. However, each session, after each stimulation, after each temporal discounting task, after 30 seconds, they would answer to a flanker task, okay, to observe if there were some carryover effects, okay? So temporal discounting task with the transcranial alternate current stimulation, 30 seconds, then the flanker task. What is it, the flanker task? It is a psychological test used to measure attention and inhibitory control. So again, conflict monitoring, okay, where participants have to answer to a central target stimulus, and the target is flanked by congruent or incongruent stimuli. For example, congruent stimuli are like this one, and people had to press the H button, okay, for example, and for the incongruent trials, it's something like that, okay? So we have the different flankers to S, but the target letter is a different one, the H, okay? So at the end, they would have strings formed by all H, strings formed by all S, or incongruent strings that are the combination between S flankers and the target H, or H flankers and the target S. And here we assess reaction times and also accuracy, but also the post-error slowing that is computed by this formula. So from the reaction times after an error, less minus the reaction times previous the error on correct trials. Okay. From these 14 participants in the stage 1, we computed the sample size by through simulation. Okay, and we through simulation, we determine that we need at least 40 participants to reach the 90% of power with Bayesian models. Okay, so we start after this stage 1 pre-registered report reached the in principle acceptance. We started with the actual data collection that followed more or less the same procedure that I told you. Okay, then there were some modifications, some minimal modifications. For example, we added that we needed some questionnaires at the end. We also added the the Barrett impulsiveness scale to assess attentional impulsiveness, motor impulsiveness, and no planification impulsiveness. Okay, for all participants, because this is a personal trait that is strongly related to conflict monitoring. So we applied generalized multi-level linear models on the temporal discounting, on reaction times, but also choices, but also on the flanker task for the reaction times, accuracies, and the post-error slowing. Okay. Also, the statistical analysis were pre-registered, and also this statistical analysis received the in principle acceptance. I must say that here the reviewers asked us to modify a little bit the statistical analysis, and this was very beneficial because actually they told us to use some techniques that we didn't knew at that time. Okay, so we actually improved our knowledge to improve before to collect the data our methodology. So after we received the in principle acceptance on February 2021, and then we started data collection, and as you can see, the timeline is very long. Okay, but you can see also that in the middle there is COVID. Okay, so that was another problem. Okay, we ended the data collection October 2022. We analyzed the data, and then we prepared the report and started the stage 2 peer review. Okay, at that time the reviewers only had to check if we followed the protocol, and we actually followed the protocol. We found results only on the temporal discounting task, so only on the task that was the online task, not and we didn't observe any carryover effect. What we found is that actually both theta and gamma tACS stimulation were able to speed up the decisions. Okay, so we found this effect only on the reaction times and only on the temporal discounting task. Okay, then we could do exploratory analysis. Exploratory analysis are analyses that were not pre-registered. Okay, it is possible to do further analysis on your data, even if you pre-registered your study. Okay, the only thing that you have to do is to clearly identify what are your exploratory analyses. So we observed that actually if we normalized our reaction times to sham, the theta tACS had stronger effects than gamma tACS only in high conflict trials. So underlining that actually it is true that theta tACS, it seems true that theta tACS has an effect, a specific effect on conflict monitoring. We also observed an association between attentional impulsiveness and choices during theta tACS stimulation. Okay, so our conclusions from this first study is that both this theta and gamma tACS can speed up decisions, but only theta can enhance the processing in high conflict scenarios. Okay, So we think that the theta stimulation is really linked to conflict monitoring, while gamma stimulation might be connected with attentional control, abstract reasoning, and working memory that could have improved the performance in that task. Okay, but we need further research to actually see if this is true. Okay, we don't observed any support for tACS influencing preferential outcomes, so we never observed differences in the choices, okay, but only in the reaction times, okay? And we also observed that there are individual differences that might modulate the theta tACS effect, especially in high conflict trials. And I mean, in this case, about attentional impulsiveness, okay? So, okay, for the study 2, I think that I can be very, very brief about study 2 because I think that it is very late. What do you think, Lorenzo? How much do we have? Should I stop here? Ten minutes. Okay, 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 okay. 10 minutes is okay. 10 minutes is okay. Okay, so study. This is with this study we only have the in principle acceptance. Okay, this is a study about transcranial static magnetic stimulation. I don't know if you ever heard about this kind. Okay, thank you, Lorenzo. I don't know if you ever heard about transcranial static magnetic stimulation because I've learned about this kind of stimulation thanks to this study. Okay. So basically, the transcranial static magnetic field stimulation is actually just the application of a magnet, okay, over the cortex, for example, 30 minutes, okay, that is not varying, okay, so it's not producing actually electric fields, but just a magnetic field. It's not like a transcranial a tMS, okay? It's just a static magnetic field. It's just putting a magnet there, okay? So there is nothing electric. It's very different from tMS, this proceeding, okay? And after 30 minutes, there are some articles that are reporting effects on the motor evoked potentials. So basically, they apply this magnetic field for 30 minutes, then they apply tMS to assess the motor evoked potentials, and but after the application of this tSMS, basically they observe reduced MEP. Okay, so this transcranial static magnetic field stimulation is able to reduce the activation, okay, of our cortices. Okay, but there are some articles that are showing that this is true, and some other articles that actually they don't find this result. So what is the truth? So we started actually doing just a meta-analysis, okay, and we just collected all the Cohen's d from all the studies. We actually used the absolute value of the Cohen's d because we were just interested in having any effect, okay? So here you can see the Cohen's d, here it's error, there is variance, and here the duration in minutes of the transcranial static magnetic field stimulation, because some of them are using 30 minutes, Some other are using only just 15 minutes, other 20 minutes, okay? So the overall effect size is a Cohen's d of 0.5, so strong one, but a very large confidence interval. If we just take into consideration effect size for the studies that applied this stimulation for 30 minutes, we have a stronger effect size, but even a larger confidence interval. However, we used this effect size to compute the sample size, but also to the next analysis. Okay. So we observed that there are high variability among the studies. For example, the dependent variable changes a lot. Okay, we have the peak-to-peak motor evoked potential amplitude in some studies, the baseline to peak motor evoked potential amplitude for other studies, or the motor evoked potential area in other studies. So 3 different dependent variables. Then they change the number of trials. Just 10 trials to evoke the motor evoked potentials. Okay, so just 10 tMS stimulation, or 20, or 30, or 40. Okay, a lot of variations also here. So also the removal of non-responding EMPs. Okay, if they are removed or not. Other variability among studies and other differences. So furthermore, in these studies, they had small sample sizes in almost all cases. So we wanted to investigate if applying this tSMS, it actually produces over the primary motor cortex, it actually produces a measurable and significant reduction on the motor evoked potential amplitude, and also to have a description of the sources of variability and confoundings in the experimental procedures and analytical processes in the verification of this hypothesis. So we want to be as robust and transparent as possible. So we used registered report and we reached the in principle acceptance, but also we want to consider all potential variants of analysis. And we used the multiverse analysis, that is a technique that is quite new. Okay, so what is a multiverse analysis? Basically, it's if you remember, before I told you that if we consider all the decisions that a researcher can take, we can have several different data sets. So let's say that this each different data set that is produced by the decision of a unique researcher is a universe. Okay? In this case, we are taking into account all the possible decisions. So we are considering a multiverse, not a universe. And we are applying the statistical analysis on all these multiverse, okay, in all these universes. And we are going to observe the results from each one of this universe, okay, to get an overall view of the effect. So this is a method that, of course, it will increase the robustness of the analysis, okay? But also it increases the transparency of the results, because it's not only our decisions, okay? But we are going to take into account all possible data sets, okay? Of course, the choices that will produce all these data sets are reasonable, are correct, okay? We are not taking into account weird decisions that just to have a different data set. It is a method that can help in pre-registering new studies if we don't know how to, which kind of analysis and which kind of choices we should pre-register. We can just take into account all of them, okay? And it is, of course, a method that considers only the reasonable choices. So only that, okay? This is a typical result from a multiverse analysis. Here you have these lines are all the possible specifications for each data set, and here each point is a different data set. And here on the y-axis, so on the x-axis, we have the different data sets, okay? Here in the y-axis, you have the estimation of the effect size, basically, of the main comparison of interest for your hypothesis. Okay, and here you can see how strong can be your effect. Of course, if I'm taking just this result, I will show that there is a statistically significant effect. But considering all the results, I can have a better view of how much robust is my result. Okay? So the procedure of this study, what we registered is that we will take the MEPs on two different baselines, one 25 minutes before the stimulation and one 10 minutes before the stimulation. Then we will apply the tSMS, effective or sham stimulation, and then we will assess the effects, the offline effects, immediately after the stimulation, after 15 minutes, or even after 30 minutes on the MEP. We consider the reviewers of the registered report asked to do a Bayesian multiverse analysis. Okay, so this is, I guess, one of the first articles that are actually using a Bayesian multiverse analysis. Okay, and we took into consideration a lot of different choices. For example, differences in dependent variable, on the transformation of our dependent variables, if we aggregate or not aggregate the data, and if we will remove pre-stimulus voluntary movements, or we will take into consideration all of them. How to check the baseline variability, okay? If we will use both baseline blocks or just the second one, okay? The phase variability, if we will use monophasic trials only or biphasic trials only. Number of trials, if we will take into account only the first 10, 20, 30, or 40 transcranial magnetic stimulation trials, if we will remove outliers or not, and how, if we will remove non-responding EMPs or not, and if we will consider control covariates or not. Okay, so we pre-registered in the peer community in here. The process was very fast. Just in 3 months we had everything accepted after very competent reviewers, and actually here the revision is not blinded, so we exactly know which are our reviewers. Okay, and this is an important aspect of open science. Okay, no blind revisions, okay, but single revisions. We received the in-principle acceptance from the peer community that can help us, after the collection of our data, to be published in some PCI-friendly journals, such as Cortex, for example, or Collabra Psychology, or other journals. Okay, there is also, I think, Nature Human Behavior that is not friendly but interested in the peer community. But there is a list in this website. Okay, because you can just check on Google peer community and you will find the website with the list of all journals that are interested on this aspect. Results, we don't know actually, because we are still collecting the data. Okay, so we don't know, but at least we have the in-principle acceptance. So if we will have the results that are saying yes, tSMS produce effects, we will be very robust because we will observe our results using all the possible decisions. And however, even if they are extremely weird results, let's say, we will get published because we have an in-principle acceptance. Okay, so a general conclusion. Of course, non-invasive brain research can improve neuroscientific knowledge, but the variety of methodologies, techniques, and statistical decisions make difficult to actually understand the quality of these findings. Open science can improve transparency, trustworthiness, and robustness of research. Pre-registrations, but more and more registered reports give the freedom not to worry about the result, and in this way we don't need to worry about not getting any statistically significant result. Furthermore, also multiverse analysis give you the freedom to show that your results are really robust, and it is also helpful in the moment that you're registering your report, because in this way you can choose not choosing directly one specific pipeline of analysis, but just choosing all possible analytical decisions that you can have analyzing this data. I remember that open science is a European mandate, okay, and it is required by Horizon 2020, but also in ERC programs and Marie Curie grants, okay? So keep in your mind that open science is the present of research, it's not only the future. You should do open science now. And I also remember again you that in February 12th and 13th of February here in Verona, there will be the next annual meeting of the Italian Reproducibility Network, and it will be extremely interesting because we will have interesting keynote speakers. Okay, so thank you, everybody. I would have really enjoyed to be with you for a spritz now, but it's not possible. So let's have a virtual spritz. Thank you very much.